Gene therapy. Introduction. Many human disease-related genes have been discovered by the medical science. Common approaches such as taking readily available drugs are ineffective in treating this disease. Gene therapy has the ability to treat or cure genetic diseases in humans. Ito yung sinasabi na sakit na nakukuha sa lahi na nadiscover ng mga expert. Tas parang yung madalas na gawin para gamutin yung sakit na yon is yung pag-iinom ng mga readily available drugs. Pero hindi naman daw effective. Pero sa gene therapy, kaya niyang gamutin yung genetic disease, yun nga po yung sakit na nakukuha sa lahi. In 2015, a group of researchers from Harvard Medical School and Boston Children's Hospital announced that they had used gene therapy to restore basic hearing in genetically deaf children. Noong 2015, ginamit daw ng mga expert yung gene therapy para gamutin yung mga batang may sakit sa pandidig or mga bingi. The study team at Boston Children's Hospital have announced that they were able to regain a higher degree of hearing down to 25 decibels, which is equal to whisper. Sinasabi na yung regain, yun yung higher degree of hearing down to 25 decibel na katumbas ng bulong.
They use an enhanced gene therapy vector known as UNtitled that was produced as the Massachusetts Eye and Ear Institute and allows for gene transfer to the inaccessible outer ear cells when delivered into cochlea. So, Massachusetts Eye and Ear Institute, or the world's largest vision and hearing research institute, focused in its International Center for Treatment and Home developed an enhanced gene therapy vector called UNtitled. UNtitled allows gene transfer to the inaccessible outer hair cells when delivered into the cochlea. This helps the hair to develop properly and help enhance the exceptional sound analysis in terms of frequency and intensity. And when the first recombinant experiments were proposed in 1971, human gene therapy became a reality. It can simply be described as inserting foreign DNA into a patient tissue in the hopes of eradicating the element. It was actually sparked by the rapid advancement of recombinant DNA technologies over the last 2 decades. So, the application of recombinant DNA technology is generally modified organisms. So, lead the advancement of agriculture, medicine, biorecommendation, and biotechnology industry. Gene therapy is the most exciting but potentially unfavorable medical area being researched without a doctor. So, in the field of research, this generation has to offer gene therapy has been one of the most exciting yet. Unfavorable medical area research without a doctor. This is because gene therapy is the one of the most complicated field in the terms of medical and research.
Basic process of gene therapy. Number 1, viral vector. Ito yung tools na ginagamit sa molecular biologists to deliver genetic materials into cell. This process can be performed inside a living organism or in cell culture. Number 2, non-viral vector. Ito naman ang methods nila ay gumagamit ng mga gawa ng tao, natural na mga compound o physical na puwersa upang mayatid ang isang piraso ng DNA sa isang cell. Gene therapy utilize the delivery of DNA into cells, which can be accomplished by a number of methods. The two major classes of methods: recombinant viruses, viral vector, recombinant vaccine, a vaccinia recombinant virus harboring the rabies virus. Glycoprotein gene has been contracted and shown to induce the synthesis of virus. Glycoprotein in infected cells and to induce rabies. Virus neutralizing antibodies and protection in susceptible animals. Example, sa mga aso, pag ikaw ay nakagat, ay kailangan bigyan ka ng inject upang hindi kumalat ang rabies. Naked DNA or DNA complex non-viral vector. The non-viral vector are naked DNA, particle-based and chemical-based. They are administered by direct administration. DNA, naked DNA, chemical, physical. Most of cardiovascular clinical trials use non-viral vectors as a mode of gene transfer. Example, operasyon. Viral vector. Viruses have evolved a way of encapsulating and delivering their genes to human cells in a pathogenic manner. Ang pathogenic ay isang medical term ng viruses, bacteria, and other terms of germs that can cause some kind of disease. Scientists have tried to harness this ability by manipulating the viral genome of remove disease-causing genes and insert therapeutic ones. Ito yung pag-aalis ng mga virus or bacteria na nag-cause ng mga sakit at ipapasok ang therapeutic upang ito ay gumaling or mag-healing.
Virus bind to their host and introduce their genetic materials into the host cell. During attachment and the penetration, that virus attaches itself to a host cell and inject its genetic materials into it. During uncoating, replication, and assembly, the viral DNA or RNA incorporates itself into the host cell's genetic material and induces it to replicate the viral genome. Next, class of strategy for gene therapy by removing the viral DNA and using the virus as a vehicle to deliver the therapeutic DNA. Although there are several strategies for gene therapy, the most commonly used method involves inserting a therapeutic gene into the genome to replace the abnormal of disease-causing gene. The gene that is inserted is delivered into a target cell via a vector. Usually, this vector is a virus. Although non-viral vectors are in development, viruses are a good choice for introducing genes into a cell because they typically operate by transferring their own genetic materials while replicating themselves. Once target cells are infected with the viral vector, the vector releases its therapeutic gene which then incorporates into the cell's DNA. The goal is that the cell will start using the new gene to make function healthy proteins. And last, The viruses used are altered to make them safe, although some risks still exist with the gene therapy. Although gene therapy is a promising treatment option for a number of diseases, including inherited disorders, some types of cancer, and a certain viral infection, the technique remains risky and is still under study to make sure that it will be safe and effective. The most common gene therapy vectors are viruses because they can recognize certain cells and carry genetic materials into the cells. Gene researchers remove the original disease-causing gene from the viruses, replacing them with the gene needed to stop diseases. This is types of virus. Many GT clinical trials rely on retroviruses or adenoviruses to deliver the desired gene. Many attempts have been made to limit the tropism of viral vectors to one of a few host cell populations. This advance would allow for the systemic administration of a relatively small amount of vector. The potential for off-target cell modification would be limited, as well as many concerns from the medical community. Most attempts to limit tropism have used chimeric envelope proteins bearing antibody fragments. These vectors show great promises for the development of magic bullet gene therapies. Next, types of viruses. Other viruses used as a vectors include adeno-associated viruses, lentiviruses, poxviruses, alphaviruses, and herpes viruses. Viruses are particularly effective form of gene delivery because the structure of the viruses prevents degradation via lysosomes of the DNA. It is delivering to the nucleus of the host cell. Viral vectors have been employed for the treatment of the virus's diseases such as metabolic, cardiovascular, muscular, hematologic, ophthalmologic, and infectious diseases and different types of cancer. Recent development in the area in immunotherapy has provided both preventive and therapeutic approaches. And the last types of viruses differ in how well they transfer genes to the cell they recognize and are able to infect, and whether they alter the cell's DNA permanently or temporarily. In addition, when viruses are used to deliver DNA to cells inside the patient's body, there is a slight chance that this DNA could unintentionally be introduced into the patient's reproductive cells. If this happens, it could produce chance that may be passed on if a patient has children after treatment.
Viral vectors. All viruses attack their host and introduce their genetic material into the host cell as part of the replication cycle. This genetic material contains basic instructions of how to produce more copies of these viruses, hijacking the body's normal production machineries to serve the needs of the virus. The host cell will carry out this instruction and produce additional copies of the virus, leading to more and more cells becoming infected. Some types of viruses actually physically insert their genes into the host genome.
This incorporates the genes of the virus among the genes of the host cell for the lifespan of that cell. Viruses like this could be used as vehicle to carry good genes into human cell. First, a scientist would remove the genes into the virus that cause disease. Then they would replace these genes with genes encoding the desired effect. For instance, insulin's production in the case of diabetics. This procedure must be done in such a way that the genes which allow the virus to insert its genome into its host genome are left intact. Mini gene therapy clinical trials rely on retroviruses or adenoviruses to deliver the desired gene. Other viruses used as vectors include adeno-associated viruses, lentiviruses, poxviruses, alphaviruses, and herpes viruses. These viruses differ in how well they transfer genes to the cell. They recognize and are able to infect, and whether they alter the cell DNA permanently or temporarily. Gene therapy is when DNA is introduced into a patient that treat a genetic disease. The new DNA usually contains a functioning gene to correct the effect of disease-causing mutation. Gene therapy uses section of DNA, usually genes, to treat or prevent disease. The DNA is carefully selected to correct the effect of a mutated gene that is causing disease. The technique was first developed in 1972 but has so far had limited success in treating human disease. Gene therapy may be a promising treatment option for some genetic disease, including muscular dystrophy and cystic fibrosis. There are two different types of gene therapy depending on which type of cell are treated. The somatic gene therapy or germline gene therapy. Viral vector are a tool commonly by molecular biologists to deliver genetic material into cells. Can be performed in vivo or in vitro. Viruses have evolved specialized molecular mechanisms to efficiently transport their genomes inside the cells they infect. Delivery of genes by a virus is termed transduction, and the infected cells are described as transduced. How is DNA transfer done? A section of DNA gene contains instruction for making a useful protein is packaged within a vector, usually a virus, bacterium, or plasmid. The vector acts as a vehicle to carry the new DNA into the cell of a patient with a genetic disease. Once inside the cell of the patient, the DNA or gene is expressed by the cell's normal machinery, leading to production of the therapeutic production protein and treatment of the patient disease.
Non-viral vector. Methods of non-viral gene delivery have also been explored, using physical carrier-free gene delivery and chemical approaches, synthetic vector-based gene delivery. Physical approaches including needle injection, electroporation, gene gun, ultrasound, hydrodynamic delivery employ a physical force that permeates the cell's membrane and facilitates intracircular gene transfer physical method. Naked DNA, the simplest method of non-viral transfections clinical trials carried out of intramuscular injections of naked DNA plasmid have occurred with some success. However, the expression has been very low in comparison to other methods of transfection. Their success, however, does not compare to that of the other methods, leading to research into more efficient methods for delivery of the naked DNA sacs as electroporation and the use of a gene gun which shots DNA-coated gold particles into the cells using high-pressure gas.
Two types of gene therapy. So I have a short definition about the two types of gene therapy. So the idea of gene therapy is based on correcting a disease at its root, fixing the abnormal genes that appear to lead to certain diseases. There are essentially two forms of gene therapy, one of which is called somatic gene therapy. Somatic gene therapy involves the manipulation of gene in cells that will helpful to the patient but not inherited to the next generation. The other form of gene therapy is called germline gene therapy, which involves the genetic modification of germ cells or the origin cells that will pass the change on the next generation.
Two types of gene therapy. Germline gene therapy. Germline therapy involves the modification of genes inside germ or gamete cells, which includes sperm or ova. Germline therapy will therefore be administered during reproductions where the modified gamete cells fuse to form a zygote. Once fused together, the zygote divides and passes on the modified gene to all other cells of the body during the development of offspring. In this way, germline therapy alters the genome of future generations to come. Although theoretically germline therapy could counteract hereditary diseases, jurisdictions in various countries such as Switzerland, Australia, and Germany prohibit the use of germline therapy due to fears on the unknown risk of this therapy and whether it causes long-term effects in future generations. Germline therapy is also extremely expensive, which further limits practical use. Somatic gene therapy. Unlike germline therapy, somatic gene therapy involves the insertion of therapeutic DNA into body cells, rather than germ cells or gametes. This means that any effects of the therapy are confined to the individual being treated and are not inherited by future offspring. The field of somatic gene therapy is surrounded by the fewer ethical issues as compared to germline gene therapy. While this may be true, this therapeutic approach remains in the early stages of development. The first hurdle in somatic gene therapy is the successful incorporation of the new gene therapy into the genome. In fact, integrating the modified gene into the wrong part of the DNA could induce rather than prevent disease. In addition to requiring the desired gene needs to be expressed, the gene expression of the new gene needs to be regulated in order to prevent overexpression that could also trigger disease.
Germline gene therapy targets the reproductive cells, meaning any changes made to the DNA will be passed on the next generation. Germline gene therapy is when DNA is transferred into the cells that produce reproductive cells, eggs, or sperm in the body. This type of therapy allows for the correction of disease-causing gene variants that are certain to be passed down from generation to generation. Example of germline gene therapy are somatic gene therapy. Is when DNA is transferred into body tissues. It especially targets cells in the body which are not passed on the person child's. Somatic cells gene therapy involves the placement of a woman gene into a living person's somatic cells. Cells that do not produce the eggs and sperm that in turn produce the next generation. Somatic cell gene therapy would aim to cure a disease only in the patient, not into the patient's descendant.
So let's answer all the questions of germline gene therapy. So this is the question number 1. Do the potential benefits of germline gene therapy outweigh the potential risk? Germline gene therapy enables the correction of disease-causing mutations that are certain to be passed on from generation to generation, sparing future generation from suffering from the disease. It enables the generation being treated to lead a healthy life. So that's the question number 1. The question number 2. Is germline gene therapy ethically acceptable? In the UK, the Gene Therapy Advisory Community was set up in 1993 to regulate the use of gene therapy. All requests to carry out any kind gene therapy on human must approved by GTAC or Gene Therapy Advisory Community, Research Ethics Community or REC, before it can go ahead. GTAC advised on the ethically acceptability of proposal for gene therapy research on humans. This regulation prevents gene therapy being used to select characteristics for non-medical purposes to design babies. So that's the question number 2. So last question about germline gene therapy. Is germline gene therapy really needed? So with the widespread use of germline gene therapy, particular genetic condition could be eliminated from the population altogether. Conditions like Huntington's disease could become diseases of the past. In the same way, the global vaccination programs have eradicated disease like polio. Germline gene therapy will correct the genetic variants of the reproductive cells of an individual, and this would be passed down to the future generation. This therapy removes a hereditary disorder from family line forever. Hereditary disorder occur at humans are possibly inherited from the germline cell. Benefits of somatic gene therapy. Somatic gene therapy replaces a faulty gene or adds a new gene in an attempt to cure disease or improve your body's ability to fight disease. Gene therapy holds promise for treating a wide range of diseases, such as cancer, heart disease, diabetes, and AIDS. Somatic gene therapy pinpoints on improvement of genetic disease by handling of non-reproductive or somatic tissues. This method of gene therapy includes the exclusion of some of the dysfunctional cells and introducing them with the clone wild type gene. So that's the benefits of somatic gene therapy. Somatic gene therapy and future generations. Generally speaking, SGT is the equivalent of standard therapy substituting bodily parts and including blood transfusions and transplants. Therefore, moral norms ruling such medical therapies are extended to this case. The equivalence is strengthened by the fact that introduce modification, which may be corrective but also ameliorative. affects only the individual subject to medical treatment is not passed on progeny. Nevertheless, it is evident that SGT, like traditional medical therapies for genetic condition, constitute a manipulation of genetic composition of future generations, enables individuals to reproduce and therefore to transmit their genetic defects to offspring. It seems that an intergenerational conflict arises between the obligation on grounds of benevolence to cure people as best as we can and the obligation on grounds of non-malevolence to avoid worsening the genetic patrimony. As a rule, the obligation of non-malevolence is thought of as being stronger than obligation of benevolence, but in the case the opposite opinion seems to prevail.
Stem cell gene therapy. Stem cell are mother cell that have the potential to become any type of the cell in the body. One of the main characteristics of stem cell is their ability to self-renew or multiply while maintaining the potential to develop into other types of cell. Stem cell that can become cell of blood, heart, bones, skin, muscle, brain, among others. So lahat ng parte ng katawan po yun, ma'am, ay pwedeng mag-develop sa stem cell para sa mga babae. There are different sources of stem cell, but all types of stem cell have same capacity to develop into multiple types of cell. So may two types po ang stem cell gene therapy. Ang una po ay the embryonic stem cell are derived from 4 or 5-day-old woman embryo that have blastocyst phase of development. The embryos are usually extra that have been created in IVF. So ano nga ba yung IVF? Into vitro fertilization. Ito po ay isang clinic where several eggs are fertilized in a test tube, then implant into woman. So ayun nga po, ma'am. Kukuha po sila ng sperm sa lalaki at i-implant po ito sa babae.
Stem cells are divided from different sources, two of which are embryonic and somatic stem cell. The somatic stem cells are the cells that exist throughout the body after embryonic development and are founded inside different types of tissue. These stem cells have been founding inside the tissue such as the brain, bone marrow, blood vessel, skeletal muscle, skin, and the liver. They remain in the non-dividing state from year until deactivated by diseases or tissue injury. This stem cell can divided or self-renew, definitely enabling to generate the range of the cell type from the originating organ or even generate them entire original organ. These generally throughout that adult somatic stem cell are limited in their ability to different bases on their tissue or origin, but there are some evidence to suggest that they can differently become other type of Coastal 2013. So embryonic and somatic stem cell, ma'am, are the cell that can generate quickly in the body, founded in the brain, bone marrow, blood vessel, skeletal muscle, skin, and liver.
The bioethics of gene therapy. Those gene therapy involves making changes to the body set of this instruction. It raises many unique ethical concerns. The ethical questions surrounding gene therapy include 1. How can good and bad uses of gene therapy be distinguished. 2. Who decides which traits are normal and which constitute a disability or disorder? 3. Will the high cost of gene therapy make it available only to the wealthy? Current gene therapy research has focused on treating individuals by targeting the therapy to body cells such as bone marrow or blood cells. This type of gene therapy cannot be passed to the person's children. Gene therapy will be targeted to egg and sperm cells, however, which will allow the inserted gene to be passed to future generations. This approach is known as Germline gene therapy. The idea of Germline gene therapy is controversial. While it will spare future generation in a family from having a particular genetic disorder, it might affect the development of a fetus in an unexpected way or have long-term side effects that are not yet known, because people who will be affected by germline gene therapy are not yet born. They can choose whether to have the treatment. Because of this ethical concern, that U.S. government does not allow federal funds to be used for research on germline gene therapy in people.
Another controversy involves the germline therapy. Discussed germline therapy is genetic modification of germ cells that will pass the change on to the next generation. There are a lot of questions since the alteration can be passed on in the United States. The government does not fund researchers on human germline gene therapy. Could the widespread use the gene therapy make society less accepting of people who are different? On a large scale, germline gene therapy could result in the selection of characteristics of approved the genetic of a population. The widespread use the germline gene therapy may make society less accepting of people who are different or who have a particular disability or genetic condition. Should people be allowed to use gene therapy to enhance basic human traits such as height, intelligence, or athletic ability? Should gene therapy be used to enhance basic human traits such as height, intelligence, or athletic ability? Should gene therapy be used?
The aspect of gene therapy. Summary. Gene therapy is designed to introduce a genetic material into cells to compensate for abnormal gene or to make a beneficial protein if a mutated gene causes a necessary protein to be faulty or missing. Gene therapy may be able to introduce a normal copy or the gene to restore the function of the protein. Summary. A gene that inserted directly into a cell usually does not function. Instead, a carrier called a vector is genetically engineered to deliver the gene. Certain viruses often use a vector because they can deliver a new gene by infecting the cell. Sample. Ang gene therapy na tinatawag ding gene transfer therapy, pagpapakilala ng isang normal na gene sa isang genome ng isang individual upang maayos ang isang pagbago na nagdudulot ng isang sakit na genetiko. Kapag ang isang normal na gene ay naipasok sa nucleus ng isang mutant cell, ang gene ay malamang na isama sa isang chromosomal site na naiiba mula sa may sira ng allele. Kahit na maaaring ayusin ang pag-mutate, ang isang bagong pag-mutate ay maaaring magresulta kung ang normal na gene ay isinasama sa isa pang functional gene. Kung papalitan ng normal na gene ang mutant allele, mayroong isang pagkakataon na ang mga transfer cell ay dumarami at makagawa ng sapat na normal na produktong gene para sa buong katawan na maiba sa hindi nasubukang phenotype.
The vector can be injected or given intravenously directly into a specific tissue in the body, where it is taken up by individual cells. Alternately, a sample of the patient cells can be removed and exposed to the vector in a laboratory settings. The cells containing the vector are then returned. Researchers must overcome many technical challenges before gene therapy will be a practical approach to treating disease. For example, scientists must find better ways to deliver genes and target them particular cells. They must also ensure that the new genes are precisely controlled by the body. What is vector? The vector can be injected or given intravenously directly to our body. All people have genes, but vector is a new type of genes to can treating disease of the patients. With genes therapy, doctors deliver a healthy copy of genes to cells inside the body. These healthy genes may replace a damaged mutated genes in active mutated genes or introduce an entirely new genes. Carriers, called vectors, transport these healthy genes into cells. This therapy will be a practical approach to treating disease. Our scientists find a simple and better ways to treat and deliver genes. When you inject of this vector or genes transferred, you need to ensure to yourself that you precisely controlled by your body.